welcome to R.K Medicos. In this section we are going to discuss about the physiology, and in physiology we are going to discuss about the nerve muscles physiology, how the nerve is going to innervate the muscles. About that we are going to see the physiological point of view, how the muscles and nerves are connected, what are the different mechanism is working to connect them. So let's start our topic. First we are going to see here about the cells of a central nervous system. What are the central nervous system cells? Do you know? It means central nervous system is basically consists of two parts, that is, one is brain and another one is a spinal cord. These are the two parts of the central nervous system. And in the brain, what are the cells are present? There are two types of cells are present in our brain. One is neuron and other is glial cells. Neurons are the functional cells of the nervous system, central nervous system. It means in our central nervous system, either brain or spinal cord, the neurons are the fiber-like structure. These are the neurons here. These neurons are the functional unit. They are going to perform the special function in our central nervous system. And glial cells are the supporting cells. They are going to support these neurons from the different regions. These are the supporting cells here. They are going to support these neurons. So the supporting cells contain 1/10 of the, it means they are content 10th part out of 10th, they are containing the 1/10 or the 10th part of the central nervous system. It means in central nervous system, when 10 cells are present, out of 10, 1 is neuron and 10 is, we can see here, 10 is glial cells and 1 is neuron. It means the number of glial cells are very high. And these are the functional cells is neuron and glial cells are the supporting cells. They are just going to support. Suppose that here is our brain. We can imagine inside these brains there is a lot of neurons are present, and these neurons are going to supported by the supporting cells here. The green color is representing your supporting cells. There is a lot of supporting cells are present, and these supporting cells are called as glial cells. And what is the functional cell here? Red one is indicating the neuron, that is the functional unit of the nervous system or the central nervous system. So this is about the cells of the CNS. Now we are going to see about the types of glial cells. These supporting cells is having two types. The one is macroglial cells and another one is microglial cells. So the two types of glial cells or the supporting cells in our central nervous system. This is the CNS. These are the supporting cells, suppose that, and inside it there are two types of supporting cells are present. Either these supporting cells are macroglial cells, or either they are microglial cells. What does it mean, microglial cells and macroglial cells? The macroglial cells are the bigger in size, but the microglial cells will be the smaller in shape and size. So the macroglia and micro means smaller, macro means bigger, glial means supporting cells. They are present in the central nervous system. So they are these three cells, that is astrocytes. They are looking like the star shape. These cells are the astrocyte cells, are the macroglial cells. Next one is oligodendrocyte cells. These cells are also the central nervous system, and these are the supporting cell. Next one is Schwann cell. Schwann cell is also the supporting cell. And microglial is the microglial cells. There is a phagocytic cell. Phagocytic cell are the microglial cells or the microglia of the brain. These are the phagocytic cell. They are the scavenger cells. They are going to phagocytosize the pathogen or the foreign bodies which are present in our central nervous system, or they are going through the circulation, they are reaching to the central nervous system. These pathogens are killing by the phagocytes in our body. So these phagocytic cells of the central nervous system is basically microglial cells or the phagocytic cell, and they are preventing the blood monocyte line. So these are the types of glial cells. They are the macro and micro. Now we are going to see about a specific one, that is the astrocyte. It is a macroglial cells. We can look on above. There is astrocytes, the macroglial cells, that the star shape. They are the star cells. There are the two types of astrocyte cells. One is fibrous astrocytes and next one is protoplasmic astrocytes. So we are going to discuss here what is astrocytes, what is their main function. So the astrocyte is a star shaped cell. There is having two types. One is protoplasmic and another one is fibrous. This fibrous one is present in the white matter of the central nervous system, but the protoplasmic is present in the gray matter. And you have to know about it. Suppose that here is the brain in the cross section. In that way we are going to cut, and we can see in the periphery, this one is the periphery of our brain. On the periphery there is a gray matter is present, and white matter in this region. White matter is here and this one is gray matter. Now you tell me where the protoplasmic astrocytes is present. So the protoplasmic astrocyte present is here in white matter, but the protoplasmic is present in the gray matter. Protoplasmic is present in the gray matter, protoplasmic astrocyte cells will be present in the gray matter, but where the fibrous astrocytes will be present? In the white matter, fibrous astrocyte. So you have to know where these cells are located. In the central nervous system, in the periphery region, there is a gray matter is present. It means there is a protoplasmic astrocytes are present. Now what is the function of these astrocyte cells? They are going to form the blood-brain barrier. What is blood-brain barrier? We discuss in the blood physiology, but for now we have to learn the blood-brain barrier is formed by these astrocyte cells. Suppose that this is the blood vessels lining. On these blood vessels lining, these star-shaped cells are adhere there, and they forming the blood-brain barrier. Through this basement membrane, the different molecules are not going to cross. They are going to check. For that reason they are called as blood-brain barrier forming cells. Next one, there is a uptake of potassium and neurotransmitter. In that area, suppose that here is a blood vessel, see this one. Inside it there is a uptake of potassium and neurotransmitter is effectively, we can see, effectively signaling the neurons. They are going to providing signals to the neuron what the potassium is going to uptake here, and the next function is blood-brain barrier. So these are the two functions of the astrocytes. They are the two types. Now we are going to see what is the main function of the oligodendrocyte cells and swan cell. These are also the type of, you can see here, these are also the type of macroglial cells, oligodendrocytes and swan cell, and here phagocytic cells. So we are going to see here oligodendrocytes and swan cell. The function is myelination. The oligodendrocytes function is myelination. Where? In the central nervous system. Oligodendrocytes going to myelinate the neuron. Suppose that this one is the neuron, this one is the cell body of neuron. This part is called as axon, and this one is terminal end. So in the axon there is a myelination is present. In the central nervous system, if this neuron is present in the CNS, then this myelination is done by oligodendrocytes. If this myelination is done in the peripheral nervous system, then it is done by swan cell. It means peripheral nervous system, it means they leave the brain and spinal cord, except the brain and spinal cord if these nerves are present anywhere in our body and they are going to myelinated. So this myelination is done by the Schwann cell basically. So it's written here, PNS myelination is done by the myelinated nerves is done by the Schwann cell in the above. Now the multiple myelination is done by basically 1 to 30 ratio for the oligodendrocytes, but in the Schwann cell they're doing 1 to 1 ratio. They are going to myelinate only 1:1 ratio, but the oligodendrocytes are multiple myelination is done by these cells. Next point we are going to see about the neurons. There are 4 types: unipolar, pseudo-unipolar, bipolar, multipolar. What does it mean polar? It means polent. We can say the unipolar is present in invertebrates. The pseudo-unipolar is present in the dorsal root ganglia. What is it meant dorsal root ganglia? Look on it. Suppose that this is our spinal cord cut section and this is our gray matter. In a spinal cord, the gray matter is situated in the center and white matter on the periphery opposite to the brain. In the brain, the peripheral region is connected with gray matter, already told you above. But in the spinal cord, the gray matter is situated in the center. And in the center, suppose that there is any sensory impulses is entered into the spinal cord from the behind, and this behind is called as dorsal part, and here is the ventral part. We can also say this is the anterior part of our spinal cord and this is our posterior part or dorsal part, through which the sensory impulses are entered in our spinal cord. Sensory impulses are entered in our spinal cord. So these sensory impulses are moving inside, and through these here a ganglion is present. These ganglion are called as dorsal root ganglion. So these ganglion are control point of the central nervous of the peripheral nervous system or central nervous system. Now we are going to see the bipolar neuron, where they are present retina and nose. In the olfactory nerves inside our nose, these type of neurons are present. It is bipolar, and the multipolar is present in the spinal motor function. They are going to leave from the here ventral region and they moving out and going to innervate the different parts of our organ of the body, and this type of nerve fibers are called as spinal motor fiber. So the spinal motor fiber is basically exit out from the spinal cord and entering inside the spinal cord is sensory. So these are the important examples we have to learn, where they are present in our body and in which region. Most important one is bipolar and pseudo-unipolar. Now look on this diagram. One point is important here. This is the structure of the neuron. We can see this is the cell body here. This is the axon. This is the cell body. And this is the terminal end or the synapse. Now you can see in the center there is a nucleus is present. Look on it here. Here is the nucleus and Nissel's bodies is present like this. Nissel's bodies. These are the basically rough endoplasmic reticulum. Next one we can see inside the cell body there is a soma. Cytoplasm is present. These are called as soma, cell body. Now here the structure, wire like a structure, the finger like a structure is coming out from the cell body or the soma part of the central nervous system. The functional unit, that is neuron, is called as dendrites, and these are going to collect the information from the adjacent neuron and sending towards the axon. It means they're collecting the impulses here and they're transferring the impulses moving inward, and this is called as forward axonal transport. when the any movement inside the neuron from cell body. Suppose that here the impulse is first time taken by the neurons by the cell body. They are going to transfer from cell body to the axon, and after that they reaching to the synapses. This type of transmission of impulses across the neuron is called as forward axonal transport of information. So the forward axonal transport is basically done from the towards the cell body to the synapses. So you have to learn it. What is it? Next point we are going to see. Now you know inside data structure what is present. Dendrites you know, cell body you know, nucleus you know, nucleus body is know. Now the point here, the cell body, this is the cell body and this is the axon. They are connecting here. This region is called as axon hillock. Axon hillock is the region where the cell body is going to connected with axon, and the initial segment is the region just after the axon hillock. This region is called as initial segment of the any neuron. We can see here this is the cell body and this is the axon and this is the terminal ends. So this region is called as here, this region is called as axon hillock connecting part, and just below it this segment is called as initial segment, the starting part of the axon. So this is important. Now next we can see there is a node of Ranvier. Suppose that here is the neuron, this is the myelinated part, this is the myelinated part, this is the myelinated part. Now look on it. You can see this myelination is done in a specific segment, and in between them there is a gap is present, and this gap is represented here. These gaps are represented here as a node of Ranvier. So the node of Ranvier is basically it is the gap which is present in between the two myelinated portion of the neuron. So it is present in the only in central nervous system or either in the peripheral nervous system, in the neurons only, and these are the myelinated portion of the neuron. This is the myelinated, and you know now the myelinated portion. So the impulses will be transmission in the jumping way, and this jumping way is called as saltatory conduction, because the information is transmitted conduction in a jumping way. That's why they are called as a saltatory conduction, because there is a myelinated area will be leads to the more transmission or the more refinement of this information. So the information are going very fast through this axon, from the axon hillock to the terminal region, and they are going. That's why these myelinated neurons are very faster to convey the information from the cell body to the terminal region. That's why we can say like this: myelinated neurons are faster conducting the impulses. They are conduct the impulses very fast. Now let's look in another way. We are going to see in the terminal end there is a small vesicles, bag like a structure is present, and these bags are filled with neurotransmitter. And here the neurotransmitter in the peripheral nervous system, basically in the parasympathetic nervous system, acetylcholine is filled with it. So in the parasympathetic system, if there any neuron is present, they are filled with acetylcholine, and it is at the synapses. So this is all about the neuron. Now you know initial body is present in the cell body region, and it is the rough endoplasmic reticulum of the RER, rough endoplasmic reticulum of the cell body. Dendrites are the receiving end. They are going to collect the information from the different part. Axon hillock is the part of cell body where the axons are originated, already told you. And the initial segment is the first part of the axon. It is the first part of the axon, already told you. The axon is going to conduct the conduction speed. The axon conduction speed is a deciding factor here due to myelination. When the myelination is done, the axon conduction facility, axon conduction speed will be increases. And if the myelin is present, the fast will be conduction. I already told you. The node of Neuron where will be the area of axon where the devoid of myelin or the absence of myelin or the no myelin. So the maximum number of sodium plus channels are present here. In the node of Ranvier, the maximum number of sodium channels are present, and it will leads to the promotes their conduction, promote the leakage of ions outside, and it will leads to the maintaining of depolarization. So it will be the helpful in the exchanging of ions and conducting the very fast. They are conducting very fast in the way, and it will leads to the depolarization and action potential transmission. Next we can see the site of action potential is initiation in motor neuron is in the initial segment of the axon. Initial segment is the first part. You know, now this is the initial segment here. In this region, the first time action potentials will be fired and moving towards the synapses. Next, we can see here nerve action potential by a graph. How we are going to represent here? Look on it. XY is y axis is representing your amplitude and x is time. Next, look on it. If we are going here, minus 90 millivolt is the resting membrane potential and minus 70 millivolt is the threshold potential. Threshold potential is the potential at which the neurons is basically going to fire in a very fast way, and the resting moment potential at which the neurons is going to be rest at that condition. So it will be the 90 millivolt. After that, these neurons are going to slightly improved their conditions and they are moving to the positive value. It means minus 85, 80, minus 75, minus 70. If the minus 70, they attain the minus 70 millivolt current in it, they will be fired automatically, and that firing will be called as something that is called as phase 1. We'll discuss it later, but for now you have to memorize it. In the nerve action potential graph, the x-axis is representing time, y-axis is amplitude. Now let's move for the first pair. Here some phases are called as, one phase name is latent period or the latent phase. What does it mean? All the phases are divided according to the ions. Ions exchange will be determining these phases. Means there is any changes which happen during the impulse transmission. Why the impulses is going to transmitted? These all are due to ions exchange basically. So in the latent period, what is happening? In the latent period is the period, it is the time gap between a stimulus and a response. It is the time gap. It means if any person is going to stimulate it, suppose it is the person it is going to stimulate it and the person is showing some response. This response is generally called as the time gap. The action is performed on the we can stimulate any person, and after that the person is showing some response, and at the time gap between the stimulus provided and response is showing, this time gap is called as latent period basically. So what is latent period here? You can see here there is the minus 90 millivolt or minus 70 millivolt. We provide the stimulus and this stimulus takes some time. And after that this stimulus will be going to fire. It means they're showing the responses. When they are showing responses, when the first phase will be started, that is called as local potential change. What does it mean? If the movement from minus 70 millivolt to minus 55 millivolt, if the charges inside the cell, from minus 70 millivolt, they are moving towards the minus 55 millivolt, it means they are moving towards the positive value. It means inside the cell some positive charges are coming, and what is it? That is sodium. Sodium is coming inside the cell and it will leads to the increasement in the positive value. It means they are coming till 55 millivolt, and this 55 millivolt at which what is happening? It is happened due to sodium influx in the less quantity. They are coming very slowly. That's why they attend this time will be taking, and that is called as latent period. There 55 millivolt is the threshold potential. It is also called as firing level for the neurons in the nervous physiology, nervous muscles physiology. The firing point of the neuron will be 55 millivolt and the resting moment potential will be minus 70 millivolt. But in the other cells, the resting moment potential will be the 90 millivolt. But in the neurons, it is opposite, or it is somehow it will be changes, that is minus 70 millivolt, and the firing or threshold potential will be the minus 55 millivolt. Now let's say in the second phase, there is a depolarizing phase will be a start. It means after the attaining 55 millivolt, inside the cells, the positive charges, what the positive charges is coming inside the cell is going to changing the potential inside the cell, and this changes inside the potential of the cell is leads to the moving towards the positive value. After the attained minus 55 millivolt, the cells will fire at a very high velocity, and we can see this moving towards the positive value very fastly, and this is due to the sodium influx very rapidly, and that is the maximum quantity of the sodium is coming inside, or the influx of the sodium is happening here, and that's why the depolarization is happen inside the cell, and the cell will be going to depolarized. Next one we are going to see in this graph, in the first phase the sodium is coming very little amount, and the cell is going to slightly above, amplitude will be increased, and after attaining the firing level, the cell is automatically going very fast to the upward, and due to the sodium is again coming inside the cell. After the attaining that level, the cell is moving downward. It means these positive charges are going out. What charges are going out basically here? That is the potassium charges is moving out, and it leads to again inside the cell what positive charges is attained. These positive charges are going to moving out. That is through the efflux of potassium. It will leads to the again cell attained negative value. They are again going for the repolarization. They are again moving towards the depolarization. So we can say here repolarization is due to potassium efflux. Next point, this action potential come back at the minus 70 millivolt. It means what the minus 70 millivolt is the resting membrane potential of the neuron. They are coming again to the minus 70 millivolt. And if this minus 70 millivolt is attained, and again this cell's potential is going again more below than the minus 70 millivolt. It means they are going more below. It means they are going more below the minus 70 millivolt. It will leads to the somehow more negative inside the cell, and this more negative inside the cell, the more negative potential inside the cell will leads to the hyperpolarization, not repolarization. It will lead to the hyperpolarization of the cell, more negative value inside the cell, and it is the 90 millivolt. That is the resting membrane potential of the any cell, and it is going to inhibit the neuron function, and due to this inhibitory neurotransmitters will be released. After the hyperpolarization inside the any cells, there is a inhibitory neurotransmitters will be released, and the inhibitory neurotransmitters is going to inhibit the special cells. What the special cell will be acting inside these neurons. So these cells are going to inhibited, and there is a inhibiting neurotransmitter is GABA. That is gamma aminobutyric acid. There is having three types: GABA A, GABA B, GABA C. So there are three types of GABA A, GABA B, GABA C. They are acting as a GABA A and C are acting together, and GABA B is acting alone through the different types of receptors. these receptors are going to perform their function by the different types of channels. Maybe the ion channels, that is chloride channels, they are performing their action. Therefore, due to the chloride influx and potassium efflux, there is a hyperpolarization is attained. How the repolarization is attained? Inside the cell, the potassium is moving out. It means the positive charges is moving out. It means inside the cells the negative charges is produced. If these positive charge are moving more than the minus 70 millivolt, is going attained minus 90, minus 80, minus 90 millivolt. It means the more negative charges will be attained inside the cell, and it leads to the hyperpolarization of the cell. So what are the elements is causing hyperpolarization of the cell? Basically, influx of chloride, it means more negative, and efflux of potassium. So these are the two elements which are responsible for the hyperpolarization. Next we can see what are the drug which is going to promote more GABA agonist. Once again, it means they are going to perform the inhibitory action, that is acting as a sedative drug. They are going to inhibit the central nervous system parts, that is barbiturates and benzodiazepine. These are the two drugs which are going to inhibit our neurons, and they are performing the inhibitory functions inside the body.